So in the last couple of chapters, we have gone through two different types of cell division. The cell cycle with mitosis, which creates more cells of the same kind. And that's important for growth of an embryo, to have more cells to make the body grow. It can be for replacement or repair of damaged or dead cells. And then the other type of cell division is called meiosis, which is very specific to creating what we call haploid cells. In animals, those are gametes, eggs and sperm, which is the first part of sexual reproduction. Of course, the second part of sexual reproduction being the fusion of two haploid cells. And in chapter 14, we looked at DNA replication, which is really the copying of the chromosomes that happens within a cell as the cell is preparing for one of these types of cell divisions. It happens in S phase in the cell cycle. It happens just before prophase 1 in meiosis. And that's just a copying of the chromosomes, just a copying. In chapter 15, we're going to look at another process that happens inside of cells. And it's maybe easy to get confused with DNA replication, because there are going to be some things that are a little bit similar, but I want to create kind of an analogy for you to compare how DNA replication, which is what you looked at in chapter 14, just the copying of chromosomes, is different than what we're going to look at in chapter 15, which is called gene expression. Okay, gene expression. And so my analogy is this. I'm going to try a couple different analogies. Let's say that you work in an office, and in the middle of the building, there's an office where the manager is. And he has walls around his office and a door. And inside the office, the manager has a copy, the only copy of the operating instructions for the company, for the business. It's a big folder with all the information on how to do everything in the company. And let's say that they decide to open another office. Maybe the business is doing really well, so they're going to open a second office, a second location. And so the manager in the office, he's going to make a copy of that big binder of all of the operating instructions. And so what he's going to do is he's just going to go to a copy machine and create a second copy. All right. And so when the new office opens, both offices will now have a copy of the operating instructions. But making a copy of the operating instructions doesn't mean they're actually reading those instructions or following those instructions. It's just making a copy. Anybody can stand in front of a copy machine and make a copy. It doesn't matter if you can understand what's written in there. So DNA replication is simply making a copy of everything so that both of the product cells gets a copy. Or in the case of meiosis, gets at least some of the information. Now, in chapter 15, we're going to talk about actually following the instructions that are in that manual. And so if you're the manager of the business, when it comes time for each department to do something, within that operating manual, there's sections. So maybe there's a section on how the accountants are supposed to do their work, and maybe there's a different section on how the sales group is supposed to do their work. And so maybe he makes copies of that section and sends it out, still within the building, but sends it out to that particular department. And they're supposed to read those instructions and follow them, actually do what's written in the instructions. So instead of just making a copy so that the new office building can have its own copy, what we're talking about in this chapter is actually following the instructions, going in and not following all the instructions at once, but reading one little section and following those instructions and actually building something, making something. That's what the DNA is. It's instructions on how to make things in the cell. So DNA replication is just copying the instructions, not following the instructions, just copy machine, copy the instructions. But gene expression is actually following the instructions and doing something, making something. And what is made in gene expression, what is built, is RNA and proteins. So the DNA is there. The DNA is the instruction manual. It stays in the nucleus. It's the only copy. Well, I guess there's two sets, but in my analogy, it's the master copy of the information, and it stays in the nucleus. And just like anything that's a master copy, you handle it carefully because it's the only copy. Let's pretend like there's no digital copies. So this is the only copy that the cell has. In my analogy, let's say it's a print copy. It's kind of like your original birth certificate. You would keep it in a protected place because it's the only one. So that the DNA, the chromosomes in the nucleus are the master copy of all the instructions on how to build things in the cell. When a cell divides, each daughter cell gets a copy. But what the cell does during other times of its life is actually follow the instructions, reads the instructions, and builds the things that are in the instructions. So that's what gene expression is, and that's what chapter 15 is about. My other analogy is I have this cookbook. And let's say this cookbook has maybe all the recipes that my mother ever made. And let's say that I wanted to make a copy and give it to my daughter. Making a copy of a cookbook isn't actually fixing meals. It's just making a copy of the cookbook. But if you follow the instructions in the cookbook, you build something, you make something, that's different. So DNA replication is just making the copy to pass on to the next cell, whereas gene expression is actually following the instructions for making one recipe. And you actually get a product from that. All right? So the product of gene expression is RNA and protein. So gene expression is sometimes called the central dogma of biology. And I should go back here. You may notice here in this picture, on the left, you have the DNA, the chromosome. They're kind of unwinding it for you. In the middle and on the far right, you have proteins. For whatever reason, they don't show any RNA in this picture, although they mention it down here at the bottom. But remember, proteins can have alpha helix. They can have beta sheets. The alpha helix is shown as the red spiral. The beta sheets are shown as a flat yellow ribbon. And then the whole protein, some parts don't have alpha helix or beta sheet, and they're just like little strings. And the whole thing is folded into a three-dimensional shape. We call that the tertiary structure of the protein. That three-dimensional shape. So you can kind of see some examples of different proteins. All right, so like I was saying, gene expression is actually how the cell reads the information on the chromosomes. And usually we think of gene expression as being just instructions for making one RNA or one protein. So when gene expression happens, it's typically thought of in an individual event of just one gene. A human chromosome has 1,000 genes on it, on average, maybe 1,200. But when we're talking about it in this chapter, we're going to be really focusing on one event of gene expression at a time. And so this is called the central dogma of biology. Dogma means like the most important central idea. And so central dogma is probably a little bit repetitive. The central dogma of biology. Everything centers on this concept, which is amazing because biology is a big thing. And the central dogma was first described by Francis Crick. Francis Crick, you may remember Watson and Crick first described the structure of DNA in 1953. And they didn't just quit science after that. They kept going and doing research and such. So they had other accomplishments. But once you win the Nobel Prize, which they did, it's hard to beat that. But Francis Crick worked on this as well in the 60s. And this picture shows the concept of gene expression. So DNA in the nucleus, the chromosomes in the nucleus, if we're talking about eukaryotic cells, or for bacteria, it's just the chromosome, has all the instructions for building everything in the cell. And in this chapter, we're going to talk about how those instructions are read and used to make something. And this is the flow of information in the cells, what we call it. The DNA in the nucleus, that's the original copy of all the instructions. And so if the manager in that business, if he's going to send instructions to the accounting department or to the sales department, he's not going to actually he's not going to rip out a page from the original manual and send it out to them. He's going to make a copy, a cheap copy, and give them the copy. He's going to keep the original in the manager's office. So in the same way, the DNA always stays in the nucleus. So a cheap copy, that's what I'm going to call it, a cheap copy of a gene, which is just one little section on one chromosome, is made. And in this language, the cheap copy is RNA. RNA is like a flimsy version of DNA. DNA is much more stable. RNA, is very short-lived. It's not a super stable molecule comparatively to DNA, but it's a cheap copy is what we're going to call it. So the instructions from one gene on one chromosome, one little spot on one chromosome will be copied into an RNA molecule. And that RNA molecule will leave the nucleus. So that cheap copy of the instructions will be passed out of the nucleus, out of the manager's office, as my analogy, and sent out to accounting. In fact, what's going to happen is the manager is going to keep making copies of those same instructions and keep sending them out. So he's kind of annoying. The manager is annoying. We can probably all relate to that, right? So he keeps sending cheap copies of the same instructions out. And the accounting department, apparently, when they get them, they follow them, but then they get destroyed. The instructions get destroyed, so the manager has to keep sending more copies so they can continuously have a fresh copy of the instructions so they can at least make some of the product. And the product here is going to be protein. So the RNA is a cheap copy of the instructions, and the protein is what's made when the instructions are actually read. So transcription is the name of the process which copies the DNA into an RNA molecule. That's called transcription. And I apologize that both of these terms start with TRANS. Transcription creates the RNA from a DNA template. And then translation is where the RNA is actually used to make something that's fundamentally different. It's not copied into protein because you can't copy RNA into protein. That doesn't really make sense. But the information in the RNA is used to build a certain kind of protein. And the protein can be the final product of this process. Now, the protein is a chain of amino acids, and it folds into its three-dimensional shape. And you'll remember that the shape of a protein, like an enzyme, is important for how it functions. If the protein gets to, if you boil that protein, it unfolds and it doesn't have that folded shape anymore, then it doesn't do its function. But normally the protein will fold as sort of an inherent, it's really like a kinetic process, a naturally occurring process of kinetics that will fold the protein into its correct shape. It's kind of amazing that it will almost always find its correct shape. And there's some detail that goes into that we're not going to get into, but the protein will fold and then it's in the cell. And you'll remember that some proteins are actually delivered into the ER, the endoplasmic reticulum, and then they can be carried by vesicles to the Golgi and then maybe released from the cell altogether. That would be secretion. So this is the kind of the part of the steps, since we've already talked about protein secretion, we're kind of backing up here and I'm kind of explaining how the protein is even produced. All right. So this is the first, the really early stage of protein secretion is how the protein is even made. So in a very general sense, you have the process of copying the DNA instructions into an RNA form, just a cheap copy, cheap paper copy. Maybe the DNA, you can think of the DNA as being on a real thick, glossy paper, and then the RNA is like some kind of flimsy, cheap paper. I don't know. But the RNA is like a crummy copy. And then that leaves the nucleus and goes out to, still inside the cell, but it goes out to the ribosome. You'll remember the ribosome is what makes the proteins. And the ribosome is going to take the information in the RNA and build the protein product for that one gene. And so this process would go on for lots of different genes within the chromosomes. So we're going to focus on just really one event of this to give you a concept of how it works. And the cell is doing this all the time. Most of the gene expression happens during the gap phases, G0, G1, and G2. G0 is a resting phase when a cell is not getting ready for division. G1 and G2, you'll recall, are part of the actual cell cycle. So these are happening all the time during those phases. All right, I'm going to take a break and we'll get to some more in the next segment.